Okay, we're back for the second day or second part of our cloning protocol. That's this cloning from cDNA protocol. So far we have done the PCR amplification of our target sequences. We have also digested our PJC 53.2 plasmid, and we've run these items on a gel to make sure that we had a band for the PCRs and that we had two distinct bands for the digested vector. So after I ran out that gel to make sure that the vector digest had worked properly, I cleaned up those digested vectors using a Zymo Clean and Concentrator kit, and there's a separate video explaining how to use that kit to purify DNA. And then I used our spectrophotometer to determine the concentration of those digested vectors. There's another video to show you how to do that as well. So at the end of that, I have 4 PCR products. They're still in the PCR tubes that I know are good enough to have strong bands. And I have 2 digested PJC 53.2 vectors. Those I eluted in water. And those have concentrations. One has a concentration of about 80 nanograms per microliter, and one has a concentration of 55 nanograms per microliter. So in the protocol, it says that you need to do a single digest of 1 microgram of PJC 53.2 for 3 or 4 cloning reactions. And actually I have a little bit, I have extra. So if you digested more than you need, you can keep that in your freezer box and use it the next time you have cloning, or if you have any redos after the first try. So you don't necessarily have to do that digest every time if you have remaining PJC that's been digested and cleaned up. So the next step is to take that digested PJC 53.2 vector. We're really interested in the part that has the promoters and the backbone. And we're going to ligate that with our PCR products. And that way we can generate this gene of interest. So I have a little diagram of this. So we're trying to generate this gene of interest cloned into the backbone with these promoters on each side. And a ligation is a enzymatic reaction in which you take DNA molecules and stick the ends of them together. And the enzyme that facilitates that reaction is called T4DNA ligase. As with the other reactions that we have talked about so far, you'll need a buffer to go with that enzyme. So proteins don't like to work in just water. They usually have a pH preference, or they might need salts or things like that in the environment. So we're going to use this buffer called rapid ligation buffer. If you've done ligations in another lab, you might have done this differently, but this is a quicker ligation that you do at room temperature just on your bench top, and it takes a shorter period of time. Other people, I've done this before where you ligate overnight at a cooler temperature, so you may have done something else in your research before. So I'm thawing out my rapid ligation buffer, RLB. You want to make sure, as with all the buffers and all the protocols, that you get things completely thawed before you start your mixing. I've gotten out my 2 digested plasmids that have been cleaned up with the Clean and Concentrator kit. I've retrieved my PCR products. And I will say that when you run your PCR, you might find that you have some strong bands and some weak bands. That's okay. As the protocol says, you can use 2 microliters of the PCR product if you have a strong band, which I did for these 4 targets, but you can look at your bands and have someone talk it through with you. You can add more than that if you need to for some of your clones. You might have 10 clones. Some of them have very strong bands, some of them have weak bands, and you'll set up the ligation differently. So in the protocol, it says to combine for your reaction 10 microliters of RLB, 70 nanograms of PJC 53.2. So I had one that came out to about 80 nanograms, so I'm just going to use about a microliter of that. It gives you a place to figure out how much you need to add of your PCR product. So 2 microliters if you have a strong band. You might add 5 or even maybe 7 if you had a really, really faint band. Sometimes you can get cloning to work even with just a tiny faint band. The T4DNA ligase, we're going to add 1 microliter, and then up to 20 microliters with sterile water. So I have gone ahead and written out my master mix ratio. This master mix is unique because in this master mix, we're not going to add the ligase to the master mix. We almost always add the enzyme to the master mix. But in this case, if we added the ligase and the buffer and the digested vector without adding the PCR product, this enzyme works so quickly and at room temperature that it would start ligating that vector back together before you would get the PCR product in there. So we'll combine the rapid ligation, buffer, the water, and the digested vector in a master mix. We'll put that into some tubes. So I've labeled both the master mix tube MM, and then I've labeled a set of tubes LIG for ligation, and then the numbers. And remember, I've put in my lab notebook what each number represents. And then these are dated and can have your initials on the side too. So once you have your rapid ligation buffer and your digested vector thawed, you can combine your reagents. So in my case, I'm going to add the RLB first. So the RLB first. I'm going to add the sterile water second. And then I'm going to add the digested vector. So this is just the PJC53 that's been cut with EAM and cleaned up. So the master mix will only have those components in it. And then because this does have a nucleic acid in it, the digested DNA, I'm not going to vortex or pipette up and down, I'm just going to flick. And then I'm going to pipette of that mixture, I'm going to pipette the correct amount into each tube. For me, this is 17 microliters because I'm going to add 2 microliters of PCR product and then 1 microliter of ligase. So I'm adding the 17 microliters of my master mix to each of my ligation tubes. That's just a small amount, so it'll look something like this. Your master mixes you'll never use, so you can always just throw out your master mix tubes. Okay, then I'm going to add my PCR product to each tube. So because I have strong bands, I'm going to add only 2 microliters of this. But as I said, if you have a weak band, you might set up your ligation differently. So I'm taking the 2 microliters of the first PCR product and putting it in the tube, and I'm just going to give it a little bit of a stir with my pipette tip. I'm not going to pipette up and down because I don't want to shear the DNA. Okay, so in these tubes now I have everything except the ligase. And as I mentioned, we're adding the enzyme last, which is kind of unusual, but this means that the ligase is not around one of the pieces of DNA until both are present. So I'm going to go grab the DNA ligase, which is in the freezer. I'm going to bring it on a cold block, and then we'll get started on adding that last reagent. So DNA ligase is an enzyme, which means we're going to keep it on a cold block from the time it comes out of the freezer to when it goes back in. And we're only going to get it out for the time that we need. And I just need to add 1 microliter of this enzyme for each of my ligations. I'm going to just pipette 1 microliter and then leave the tube on the cold block while I pipette it into my ligation. Give it a little bit of a stir like that. And then repeat the process for the other 3 tubes. It's okay if you get a little bubble in there. Just make sure, because you're adding small volumes of the DNA from your PCR and the ligation, that you're really looking at the pipette to make sure you're transferring the enzyme. Okay, so now I'm going to cap this enzyme. I'm going to put it back in the freezer right away. And my ligations are actually ready to go now that they've been stirred. So you can set a timer. I usually count up just so it doesn't beep at me. And this needs to incubate for over 30 minutes. If I can do it, I like to let these incubate for 2 hours at room temperature because it works better that way. But if you're in a pinch for time, you might try 30 minutes or an hour or something like that. And these tubes can just sit at room temperature. You're going to, of course, put the enzyme away first. And then your digested vector, RLB, and PCR products can go back into your freezer box. So after your ligation is complete, the next step is to transform this ligation into bacteria. And what will happen when we do this transformation is that the circular plasmid that contains the backbone of the vector and your gene of interest will be taken up by a bacteria and then amplified as that bacteria replicates itself. That way you can purify that amplified plasmid in order to sequence it and make sure that the cloning is correct, and also to use that purified plasmid for generation of a PCR product for double-stranded RNA and riboprobes, which are two of the main things that we use this clone for. So when we're doing this transformation, which is the next step on the protocol, you'll be working with E. coli bacteria that are chemically competent. That means they've been subjected to specific chemicals that prepare them to take up the vector and to then replicate with this vector inside it. These chemically competent cells are in the -80 freezer, and they need to be kept cold. So the first thing you'll need to do is get a little ice bucket. There's an ice machine in the middle of the 6th floor of our building. So however many samples you have, so I have 4, that's how many aliquots of bacteria you will need to pull from the freezer. And these are already aliquoted into 50 microliter aliquots, which is exactly the amount that you'll need for this protocol. If you've never worked with E. coli before, it's probably wise to have someone show you these next few steps by yourself. E. coli is a bacteria that is not extremely harmful, but it is still a biohazard, and so you need to be trained properly before you can do this yourself. However, I'm going to show you how to do this, that way you'll be able to kind of speed things along. So the first thing I need to do is go get my tubes of bacteria and put them on ice. This will allow them to thaw. I'm at about 10 minutes before I'm ready to deal with my ligations. So I'm going to open up this -80 freezer right here. And there on the bottom shelf is a styrofoam box. And if you pull it out, I'm having a hard time doing it with one hand. There's some aliquots of bacteria. Use my hand. So you would not be holding a camera, and this will be a little easier for you, but I have 4 of these little tubes that I'll put on ice. You're gonna make sure always to close the -80 freezer really well. And then what I do is take these tubes and just put them on ice and allow them to sit for a few minutes to thaw. And then we will resume our transformation. So these bacteria will thaw even though you put them on ice as they change temperature from -80 to just around the melting point of ice. They will still be cloudy, so I think you can kind of see that they're cloudy even after they've thawed, so you shouldn't wait till they look like water or anything like that. The other thing I've done with these tubes is to number them so that I have them labeled for my ligation. So ligation 1 will be 1 and so on. You may find that you need to wipe off the lid of the tube with a chem wipe because there's some condensation that might make it hard to write on until the tube is dry. The other thing I've done is I've grabbed one of these biohazard buckets. It has a lid, and I just kind of put the lid a little bit ajar. That way I can eject my tips into the biohazard when I'm done. So I'm going to take 5 microliters of my ligation and pipette them into the competent cells, and I'm just going to give them a little stir with my pipette tip. You can also flick a little, and then eject that tip into the biohazard bag, and I'll do the same thing for the other 3 ligations that I have. These ligations can be stored at -20 for a period of time. I almost never go back to the ligations. If my ligations don't work, I usually just redo them. But when you're just starting out, just in case you have some kind of a problem with your transformation, you may want to keep them overnight or until such time as you know that the transformations have worked. So I'm also just kind of flicking these to mix them. And these bacteria are treated with chemicals to make them prepared for taking up the DNA, but this makes them very fragile. So this entire incubation will occur on ice. So I have these tubes and I just put the ligation into the bacterial tube and put them on ice, and they can stay that way for 30 minutes. You can let this go a little bit longer if you need to. So say you were going to go have lunch or something. I think even up to an hour is probably fine, but you don't want to let this go overnight or over an hour. So you'll let them incubate on ice. Next, we will recover the bacteria to make the cells healthy again. We'll first heat shock them to get the DNA into them, then recover, and then we're going to plate them. So one thing I always recommend you do when you've started your transformation is to go ahead and go get out your agar plates. The agar plates are made out of LB with kanamycin, and kanamycin is an antibiotic, and this backbone has kanamycin resistance. So if there is a plasmid that has that backbone, it should allow the bacteria to grow on can-containing plates. If you'll recall, I earlier said the cCDB gene, which is over here, is toxic to bacteria. So if you had any undigested plasmid and that got transformed, that would kill the bacteria, but the only thing that should survive is something with this backbone that has can resistance and your gene of interest. So we have some can-containing agar plates made out of LB, which are in the refrigerator. I'll show you where those are. They're in this fridge that says EWR, and they are back in the back, kind of in the back corner. So I'm going to get out one of these plates per sample and just put them at room temperature to acclimate. Sometimes they can be a little bit wet and have condensation, and allowing them to come up to room temperature will help you when you're ready to plate your bacteria. So I'm at the end of the 30-minute incubation of my transformation reactions. And after a transformation, after this ice incubation, the next thing we're going to do is do a heat shock for 90 seconds at 42 degrees. So there's a water bath that's usually set to around 42 degrees here. And I'm going to place my samples. Here's the one on the right, it's at 42. I'm going to place my samples in a little rack like this and drop it into this water bath and set a timer for 90 seconds. So as I mentioned, this incubation and heat shock of the bacteria helps them to take up the DNA that is in their environment and bring that into the cells. It is the combination of the chemicals that they're in to make them competent, and the heat shock will achieve the transformation. So after this step, what we're going to do is incubate them for 2 minutes on ice, and that just cools them back down. We're going to add 1 milliliter of LB medium, which is a growth medium for the bacteria, to the tubes. And then I'm going to incubate them at 37 degrees in the shaker for an hour. And this period is a recovery period, which allows the bacteria to reacclimate to healthy media, heal the damage that's been done with the heat shock and the chemical exposure. And this allows the bacteria to be really healthy at the time that you plate them on the agar plates with kanamycin, so you get more survival of the bacteria that have taken up your plasmid of interest. So if you skip this recovery step, it might still work, but you'd expect to have fewer colonies of bacteria for each transformation than with the recovery step. So we usually include it for ligations. So I'm now at 1 minute and 30 seconds. So I'm going to take this out and put these samples back on ice and restart my timer for 2 minutes. And in just a moment, I'll resume the video and show you how to add the LB and set these reactions up to shake. So after 2 minutes, you're going to add LB media to each tube. LB media can be found, you can make it, but it can also be found in bottles. It's a little bit yellowish in color. I usually pour some of this media into a conical tube. That way, if I contaminate the LB media, I've only lost, maybe a few milliliters instead of that whole bottle. If it's contaminated, it can't be used. So I'm going to add 1 milliliter, of this media to each tube. You can use either one tip if you're extremely careful, or you can replace your tip for each tube. Just make sure that it doesn't touch the bacteria or the tubes, and then you should be fine. And I'm putting them back in the ice once I put the LB media in there, just for the sake of keeping them organized. And then once I'm done with this tip, because there's a possibility that it's been exposed to bacteria, I'm going to put it in my biohazard waste. So I'm taking these samples, adding the LB media to them. That one I'm touching, so I'm going to discard that tip. And then I'll have one more to do. So now again, we're going to incubate these for 1 hour. What I typically do is place these in the shaking incubator that we have in the lab. And I used to just tape these down into the floor of the incubator, but one time we tried that and someone's samples got lost in the floor of the incubator. So what I do now is take some tape, I put these samples, kind of adhere them to the tape, and then I have an empty tip box just like this, taken out the tray, and I tape my samples into the bottom of the tip box. So this ice can just be thrown in the sink when you're finished. So I've put my samples, taped them to the bottom of this box, and then I'm going to take this box with samples into the other room where there's a shaking incubator. And this is the shaking incubator right here. And there's buttons on the side over here where you can turn off and on the shaker or the heat if you need to. And I'm basically going to shaken for 1 hour, and then we will resume the video. So I have taken my bacterial tubes out of the incubator, and I'm over here at the centrifuge because I'm going to spin them down for 1 minute. Room temperature or cold is fine at 7,000 RPMs. The reason I'm going to do this is my samples are now in 1 milliliter of media, and that's so much liquid that it will not absorb onto the agar plates. And I'll take you over here to the rest of my setup. I have set up a vacuum flask. This is attached to the vacuum, which is on, and this is also attached to a little syringe which has a just a P200 pipe tip on the end so that I can remove the extra supernatant from my tubes. I also have set up one of these LBK plates for each one of my samples, and I've labeled them, and I have my P200 tips and my P200 pipette here with my biohazard container. And lastly, I have this set up that is our Bunsen burner. I have a beaker with 100% ethanol there, and I have this Bunsen burner that's plugged in back here, and then also has a pedal at the bottom to light the Bunsen burner. And I'm going to turn this on, and I'm also going to tighten this to the on position. So what you should be able to see once both of those things happen is that when I push this pedal, which I think I have upside down, sorry, you should get a flame. Okay. So the first thing I'm going to do, I have my tubes out of the centrifuge. What you'll be able to see is that the liquid will be more clear, and you should see a little white pellet right in the tip of the tube. And if you look at the gradations on the side, there's a little line that says 0.1. I'm actually going to vacuum aspirate until I have about 50 microliters, so that's halfway between 0.1 and the bottom. So I have my vacuum aspirator here, and I have this little tip on the end of the vacuum manifold, and then I'm going to hold my tube like this and vacuum against the side until I only have about 50 microliters left, which is like just a little bit. I'm going to do this for each of my samples. Make sure you don't aspirate that pellet, or you will have to start over again. So the pellet is the bacteria. We're just removing this extra liquid so that we can plate the bacteria. Now your vacuum flask will now have bacterial solution in it, so this needs to be bleached when you're finished for 20 or 30 minutes, and then it can be rinsed out with water. Okay, the next thing I'm going to do is take my pipette and resuspend those pellets, so just pipetting up and down. So I'm just pipetting up and down. Now you can see the solution becomes cloudy again as those bacteria re-enter solution. And I'm going to pipette this just onto the plate. I'm not going to stab into the plate, just let the droplets fall onto the plate. I'm going to put both the tube and the tip in the biohazard trash as I go along. So I'm going to do this for each one of my samples so that I have little kind of droplets of bacteria on each plate. If I had a lot of samples, I have 4, so I'm going to do all 4 of them. If I had maybe, 10 or so or more, I would do this in little batches. So I might vacuum, aspirate most of the solution, but only pipette them onto the plates and plate the bacteria maybe 4 or 5 at a time. So I've now placed the bacteria. Each one of my LBK plates has a little blob of bacteria on them. And so the last thing I'm going to do is spread these cells. So I have my beaker with the ethanol in it, which I'm going to remove the cap from. There's a cell spreader in there, and I'm going to run this just briefly across the flame, being careful not to let it drip too far. This will catch the ethanol on fire. I'm going to wait till that flame goes out. I only, push the floor pedal long enough to get a flame to light the cell spreader, and then I let go of the foot pedal so that it's not on all the time, wasting butane. And so you can see that there's some liquid on this. I'm just going to gently spread these cells around with the cell spreader. Some people try to push too hard or put this on the top of the bench, and you can really gouge the agar because it's pretty soft. If you hold it like this, you can sort of, I think, avoid that a little bit. Sorry, that vacuum is making a noise. So once the liquid is fully absorbed, you should be able to tell that just by looking at it. So if you can still see little liquid droplets, you may keep going. The other thing you can do is if the cells don't entirely spread, as in the liquid is still there a little, you can let them sit face up for a few minutes on the bench top to let some of the water that's in the solution evaporate. So I'm going to do this again. I'm dipping my cell spreader after I'm finished into the ethanol. I'm going to run the cell spreader against the edge to kind of get all the extra ethanol off. I'm gonna light my Bunsen burner, light my cell spreader, wait till it's not flaming anymore, and then spread the cells for my second plate. One thing that can occasionally happen if you're being careless with this is that you can accidentally light your beaker of ethanol on fire. And if you have the kind of presence of mind, what you can do is just drop the lid. There's this little petri dish cover onto the beaker. This would deprive the flame of oxygen and put the fire right out. What you really don't want to do is overreact, especially to knock this over and then you have flaming ethanol everywhere, which I've seen happen once. You really just want to try to find this or something like that to cover it up. And like I said, that I've done that before by accident, and as long as you have the presence of mind to cover it up, it's really not very dangerous. The flame, even though it's not specifically sciencey, is one of the things in the lab that can hurt you. So in addition to being careful when you're doing this, and I'm just going to keep going with the other two plates, you want to probably do this when someone else is in the lab, just in case you have an accident. You also want to make sure before you start that you know where the fire extinguishers are, just in case, and where the showers are, just in case. And those are the things that you could use to put out a fire if it gets on your clothes or if it gets on your bench in a very worst case scenario. Like I said, I've only seen that happen maybe a couple of times, and those were times when someone, like I said, was being really careless, dumped over an ethanol bottle, and even though you've probably used a Bunsen burner before, just remember that Bunsen burners are flames just like any other fire, and you need to be very careful with them. So I would think about, how you set things up depending on if you're right or left-handed. You don't want to have this Bunsen burner on a cluttered bench. You don't want to have paper around. You don't want to have the biohazard bag really close to the flame so that it could catch on fire. You really want to have the Bunsen burner alone and far from you and anything flammable on your bench. Having this foot pedal also does help because you don't have the flame continuously going, and that can avoid some just accidental run-ins with the flame itself. Once you're finished, you're going to push the power button until the green light goes off. Just takes a few seconds. You're also going to, there's a knob at the top up here. You're going to move the knob toward the off arrow until it can't move any further, and this kind of decouples the butane from the Bunsen burner, and then you can unplug it. And this will just keep things from someone from accidentally stepping on the pedal or something like that when this is not in use. After you're done, you'll have your samples of your bacteria on your plates. There's an incubator that I will go show you. It's in the other bay. So this is the bay with the common freezer in it. And under this bench, which is Tulip's bench right now, there is an incubator set at 37 degrees, although the temperature is a little bit off at the bottom. It's pretty old. And so you can just put that in the incubator. And these bacteria need to grow overnight. In the morning, you should see some colonies, which are tiny little bacterial growths on your plates. If you plate your cells and you can't come in the next day, make sure that someone else can get those bacterial plates out for you. And those plates can be put in the refrigerator at 4 degrees overnight. So for example, I can't tend to these tomorrow, so someone's going to take those plates out for me and put them in the fridge, and then when I come in, I'll look at them and proceed with the protocol. To clean up, you just need to make sure the Bunsen burner and the ethanol beaker and the biohazard basket are all put away. You may use these on the common bench, in which case you can just make sure they're all straightened up. You'll take your tips and pipette back to your bench, and then you will rinse out this vacuum flask with some ethanol, let it sit for 20 or 30 minutes, and then rinse it out with water. And it stays behind the sink in the close side of the lab. So then it will be ready for the next person. The tip that was on here can go just in the biohazard trash too. All right, so hopefully we'll get some colonies from these cloning reactions and transformations, and we'll get to move forward with the protocol.